So we ended off class talking a little bit about methylation and acetylation. We watched a video about the supercoiling of DNA, which is in the black here, around these purple proteins here called histones. And what histones do is they just allow DNA to pack more tightly because it wouldn't be able to actually fit in the nucleus if it wasn't at least somewhat wrapped. It supercoils during mitosis and meiosis, or cell division. But it's generally always coiled, and this coiling can be influenced by either by methylation and acetylation. As you see, during the acetylation, it allows parts of the DNA to be shown so that the information on it can be expressed. And over here, the methylation actually silences the DNA. The DNA is so wrapped up and folded and supercoiled that the information on the DNA, these black pieces, isn't actually accessible for expression, gene expression. Expression just means making stuff from the DNA. So let's move a little bit closer to about what DNA is. This is the general form of DNA, this being one strand here and this being another strand here. The backbone of DNA is made of sugar phosphates, and what connects via hydrogen bonds the two strands together are called nitrogenous bases, and they are connected by hydrogen bonds. If we move along here, there's two examples of DNA and RNA. One is deoxyribose nucleic acid, and the other is ribose nucleic acid. Deoxyribose is a sugar. It's a five-sided ring structure. It's called deoxy without oxygen because the second carbon here doesn't have an oxygen. As you look over on RNA, ribose nucleic acid, on the second carbon it does have that hydroxyl group. So this is called ribose, which is the sugar, and this sugar here is called deoxyribose. But all DNA and RNA have nucleotides have three major parts. They have their sugar, they have the phosphate group attached to the number 5 carbon, number 6 carbon actually, 1, 2, 3, 4, 5, number 5 carbon, and they have a nitrogenous base here. All right, so sugar, phosphate, nitrogenous base, the sugar attached to the number 5 carbon. The phosphate group obviously is a polar, hydrophilic, and charged group. How do DNA and RNA differ? DNA is always two strands, always, even if it's circular like in a bacteria. RNA is almost always single strand. It may occur as a double strand once in a while in living organisms, but it's generally a single-stranded thing. The nucleotides that occur in RNA are cytosine, guanine, adenine, and uracil. Two of them are purines, double-ringed structures, and two of them, cytosine and uracil, are pyrimidines, single-ringed nitrogenous bases. And then if we go over to DNA of cytosine, guanine, adenine, and thymine, the two double-ringed purines are guanine and adenine, and the two single-ringed pyrimidines are cytosine and thymine. Cytosine is always going to bond with guanine, and adenine is always going to bond with thymine, unless it's bonding hydrogen bonding to a nitrogenous base in RNA, then adenine will bond with uracil. So remember, A goes with T and G goes with C. A purine will always match with a pyrimidine, and a pyrimidine will always match with a purine. So those are some differences there. If we go to the more polymer structure, when we go back here, we're looking at, this is a monomer of DNA. This is a nucleotide. Whereas this here is also a nucleoside for ribosome, for RNA. Both of them are monomers, though. If we put those together, we'll get polymers of DNA, long strands of many different nucleic acids making up the entire strand. Now, if we look here, a couple of really important things about DNA are that one of the strands on DNA is always going to be going in one direction with the ribose and the phosphate facing downwards or upside down. And the other strand that's matching with that will always have the other orientation. It will be facing up. Okay. One thing to notice here, and it'll probably come back very slowly, is that one end of the DNA strand on one side is a 3' and the other end is a 5'. If you notice the 5' side, on both sides is always going to have the phosphate sticking out, and the 3' side is always going to have the hydroxyl sticking out. So if you need to identify the 3' side, you're always going to look for the hydroxyl sticking out. If you know to identify the 5' end, you're always going to look for the phosphate hanging out. Now, if you know what the 5' end is, you know where the phosphate group is going to be sticking out. If you notice that the nitrogenous bases are still bonded via hydrogen bonds, a purine is always bonded to a pyrimidine. All right? 2 purines will never be bonded to one another via hydrogen bonds, and 2 purines will never be bonded to one another. So a couple of the important things to get from here are that each strand is anti-parallel. Okay, one strand is running in one direction with its phosphate hanging out this side. The other strand will be running in the opposite direction with its corresponding 3' side hanging off on one end. So there's a 3', 5'. You'll understand why this is important later on, is because certain replication enzymes that are going to replicate DNA, can actually only move and read in specific directions. Mostly, they can only read 3 to 5. So they'll be moving up here, they're moving down here. So this is where it gets important. The base pairing rules again, adenine in DNA, adenine will always pair with thymine, and guanine will always pair with cytosine, as you notice here. In RNA, on the other hand, anytime you see an adenine, you'll see a uracil in place of thymine. You won't see thymine and uracil. If you need to identify the difference between DNA and RNA, one of the first things you should look at is the presence of thymine and uracil, because thymine will not be present in RNA, but it will be present in DNA. If you see uracil, you know it's not DNA. And if you see uracil, it's more likely to be a double-stranded RNA. These are what the purines generally look like, and these are the pyrimidines generally look like. You don't have to know anything specific about them except what they are, how many ring structures they have, and what they bond with. Okay. Remember, purines are adenine, guanine. For me, I always remember pure ag. If I remember pure ag, I'll know what the purines are, and then the rest of them are pyrimidines. So that's just a little helpful hint for you. Again, nucleotide polarity. Okay. The 5' end is the phosphate sticking out at one end, and the 3' end is where the hydroxyl is sticking out. If we were to go back again, here, you'll know this is the 3' hydroxyl end, and the phosphate is the 5' end. No matter how many nucleotides you put on the side in this orientation, which is the only way the next nucleotide will fit, you're always going to have this will always be the 5' end. On this orientation here, this will always be the 5' end, because every nucleotide you put after this will always have its phosphate sticking out, and this will always be the 3' end. Okay, so 3', 5'. We call it polarity of the DNA molecule, because polar means different ends. Doesn't have to mean positive or negative, like the water molecule. Okay. More, we're talking about nitrogenous bases in the 5', 3' end. You don't have to know what kind of bonds link them together, as long as you know what 3 to 5 means. Now, another important thing here is that actually each nucleotide has a 3 to 5 end. 3, 5, 3, 5, 3, 5, 3, 5, 3, 5, 3, 5. Okay. Over here too, 3 to 5, 3 to 5, 3 to 5. So even though the entire strand has 3 to 5, each nucleotide also has a 3 to 5' end. Okay. Now, what do these nucleotide nitrogenous base pairing rules mean? Well, the DNA nitrogenous bases, we're going to go back here. The DNA, this is DNA here. Let's say we have TGA. Okay? Well, DNA doesn't do anything really, it just stores information. We know from our past biological experience that DNA makes RNA and RNA makes protein. All right? Well, let's say we read this piece here, TGA. If we were to read TGA, its corresponding RNA piece is going to be what? If you guessed ACU, then you'd be right. So the corresponding piece of RNA that is made from this 3 nucleotide piece of DNA is going to be A, C, and U. Okay, if we were to make RNA from it. Now, if we go over to here, if you look at ACU, A, C, U, that information is going to be manifested as the amino acid threonine. Okay. Now we're going to get into what all that means later, but the important thing to understand is that these letters here, these nitrogenous bases that are associated with the sugar phosphates. The sugar phosphates are only important for the backbone of the DNA, but the real information in the DNA, the real stuff that codes for things that matter in your life, is actually in these nitrogenous bases here. All right, and they're read in groups of 3, which is what this chart here will show. Okay. Soon, I'm sure we'll get into DNA replication, transcription, translation. I'm sure we're also going a little bit faster than we would have in biology. That's probably because we've already learned it, number 1. And number 2, we've got some cool stuff to talk about when it comes to human genetics. And getting a hold of DNA and RNA and what makes it and why it is the way it is are really important to understanding replication, transcription, and translation, which will then take us to the more fun parts of human genetics. So thanks a lot for listening to the biochemistry of a nucleotide, DNA and RNA, and there will be a worksheet to go along with this, and also please, if it's stuff that's new to you, please take notes. This PowerPoint is uploaded under Schoology, so you'll have access to this PowerPoint. Thank you.